research funding from Advanced Bionics, Cochlear and Merel. I'm also a consultant for a startup hearing aid company that has nothing to do with this presentation. I'm sorry, it's not moving forwards. Okay, now it is. And I want to acknowledge support from NIH and ADCD and to numerous collaborators, both inside and outside NYU. I'd like to mention the NYU Cochlear Implant Center. It's an essential part of this research, including co-directors Bill Shapiro and Shannon McMenemy. The annual studies are led by Rob Fromke and carried out by trainees who are members of both our labs. Julia King, Aaron Glennon, and these days Ariel Haidt and Yuko Tamaoki. The two basic questions we're trying to address today are these. What does a cochlear implant sound like? And how plastic is the human brain? More specifically, how plastic it is in response to a modified peripheral frequency to place map. The translational question is, what can we do clinically when brain plasticity is not enough? And I'm going to discuss three ways to address that problem. Fix the frequency, fix the place, or hack the brain. And I'm going to explain each one of those. What does a cochlear implant sound like? Many of you may have asked patients, so what does it sound like? If patients are postlingually deaf, and I want to say that this whole talk is about postlingually deaf patients, patients who developed English language based on their acoustic hearing, then lost it, then got a cochlear implant. These are real comments from social media, where one patient with a CI asks, has everyone had to deal with the Darth Vader Mickey Mouse voices? And these responses summarize much of the talk. One of them says, I'm 9 months post-activation, I still hear them. This one says, I heard Minnie Mouse for 2 months, then it cleared up. This one never heard Darth, but Minnie and I are best friends. Why might that happen? Well, we know that in the all mammalian cochleas, there's a frequency to place function. And here in this representation of a cochlea, we show in blue the characteristic frequencies of the neurons that are presumably stimulated by a cochlear implant. These are based on Greenwood's equation. Now, when we put a cochlear implant, these are real numbers from an old Advanced Bionics device. The cochlear implant doesn't extend all the way to the tip of the cochlea, of course. So that means that the most apical electrode, say this one, that stimulates in response to frequencies between 250 and 500 hertz, may be stimulating neurons that are associated with a much higher frequency, maybe 1 or 2 octaves higher. So that's what we call anatomical frequency mismatch. Anatomical frequency mismatch is something that's fixed. It's the discrepancy between the anatomical frequency place function in the human cochlea and the normally different frequency place function that's imposed by a cochlear implant. That function depends on the frequency table in the patient's processor and on the exact electrode location. I think it's important to distinguish that anatomical frequency mismatch from perceptual frequency mismatch. So this is the discrepancy between the pitch percept elicited by a tone in a normal ear and by the same tone in a cochlear implant ear. The two concepts are related, but they're different. Anatomical frequency mismatch does not change. That depends on how the speech process is programmed and where the electrodes are. Perceptual frequency mismatch may change if the brain's patient is plastic, because the pitch percept elicited by a given tone in a ear may change over time. And in the first study, we're going to see how we might measure that. Now, what we use as the anatomical frequency place function in the cochlea has traditionally been the Greenwood function. And this has been taken as the ground truth by our field, and that is, I believe, quite wrong. Why do I say this? Because the Greenwood function is not based on any physiological data. It's based only on mechanical data, movement of the basilar membrane. These data were obtained from cadaveric cochleas, not from live humans, with presentation levels above 120 dB. And I recommend reading the Bekesy studies. He received the Nobel Prize for his research. They're amazingly interesting and ingenious. But the data are noisy. Data from the two Bekesy studies differ by over 50% from each other at different frequencies, and they differ from Greenwood's equation by as much as 60%. The other data set, Kringle-Bottom et al. From 78, displacement of vessel membrane has two different peaks, and the authors decided that the closest one to the Greenwood function should be the one that's been used. Bottom line, the data in support of the Greenwood function is extremely noisy and not very accurate. And it's interesting that despite these points, we as a field and industry has heavily invested in approaches that rely on this equation, and we have incorrectly accepted as the precise ground truth. I say this because in a study we published last year, Dr. Elad Sagi and I examined all animal studies that try to actually measure characteristic frequency and how that changes with presentation level. Because one very consistent finding is that the frequency place function is a certain function at threshold, but a different one at the normal loudness levels that we use in everyday life. And what we found, so what we show here in the y-axis is insertion angle, and the x-axis frequency. And the green line you see here. Oh, by the way, can you see my mouse?
Yes, we could see your mouse.
Ah, okay, good. So this green line here is the Greenwood function. And in black, we show how the function is modified when we use normal moderate loudness presentation levels. And we find that there's a discrepancy between 1/3 to 1/2 of an octave. This means that frequency mismatch that we talk about so much, the anatomical frequency mismatch, may be 1/2 an octave less than we think. And this is my collaborator, Elad Sagi, who had the leading role in this study. Now, the bottom line is that despite this correction to the Greenwood function, typical locations of CI electrodes still result in an anatomical frequency mismatch, not as much as we used to think. Now, what about perceptual frequency mismatch? I'm going to show you one way to measure it, which is a novel speech-based measure. What we do here is present a sentence to a patient through the speech processor. Then there's a brief silence and present the same sentence through an acoustic model to the acoustic ear. Importantly, the patient is asked to adjust the acoustic model until the sentence sounds as similar as possible in the CI ear and the acoustic ear. This is the graphical user interface that patients see. They select one of these squares in this grid, and they move this slider from 0 to 5. So each square represents a different acoustic model. So I can, as you move up or down, I should say that these acoustic models have the same number of channels and the same analysis filters as in the subject CI map. What we're changing is the outputs, tones, or noise bands in the vocoder. As we move up or down, we change the low frequency edge of those output tones or noise bands. As we move left, right, we change the high frequency edge. As you can see, the low frequency can be selected anywhere between 63 and 1800 hertz, the high frequency edge between 3000 and 19,000. The slider determines the amount of overlap between noise bands. 0 represents tones, 1 represents separate noise bands, 2, 4 represent more and more overlap. Let me play some of these so you can get a sense. This is, so this example would be an acoustic model where the outputs noise bands go from 1200 to 5000 hertz. So a lot of frequency mismatch, especially in the low frequency. So that's very hard to understand. Let me show you one example with less frequency mismatch. And one with no mismatch at all. The sentence is the player lost a shoe, of course. And you can tell how the sentence, the model without frequency mismatch can be quite intelligible. And even the one with some mismatch can be intelligible too, even if the quality is bizarre. Mahana Azadpour has had a significant role in developing this application. So we tested 54 subjects with single-sided deafness. Those are required for this experiment. And they had different amount of experience at each testing session. They were mostly cochlear and metal users. There were also some AB users. And these are some of the results. Each one of these thin bars shows the range of frequencies for the output tones or noise bands selected by each subject. Here to the left is cochlear, in the middle metal, in the right advanced bionics. This big yellow bar shows the frequency range of the analysis filters used as a default in standard of care for cochlear, 188 to 7,938 hertz. So if somebody selects output tones that sound exactly like this frequency range, we say that this person has no frequency mismatch. What we see is that at the low frequency edge, the patient selection was sometimes as low as the analysis filters, but more often than not, it was higher, both for cochlear and to some extent for metal. So this is consistent with the fact that the electrodes don't extend all the way to the tip of the cochlea. This gives us a chance to measure frequency mismatch. At the high frequency edge, data were a lot noisier. And I think there's a reason for that, which we don't have time to discuss. So the conversation that follows focuses on the low frequency edge. The low frequency edge gives us a chance to quantify perceptual frequency mismatch. This patient selected output tones or noise bands that start at 1600 hertz. I'm sorry, 1788. So the difference between 188 and what the patient selected is 1600 hertz. This patient selected a lower frequency edge to begin his output noise bands. This one even lower. Let me play some of these so you can see what these sound like. This one is very hard to understand. I can't see your faces, but I'm willing to bet none of you understood that. This is still hard. This one may be a bit more intelligible. No, I think I played the same. Let me play the one without frequency mismatch.
Try to stay focused on the task at hand.
So obviously, if you had the choice to hear from a cochlear implant like this instead of like this, that would be much better. And so in response to the question, what does a cochlear implant sound like? There isn't a single answer. There are as many answers as there are listeners, but a very clear common trend. The self-selected acoustic models tend to require output tones or noise bands whose low frequency edge is higher than the analysis filter bank. And I say this because all these bars start higher than the cochlear standard FAT, and most of these start higher than the standard metal FAT, and so for metal. So this means there is evidence of what we call baselward shift, and it's consistent with studies of electroacoustic pitch matching. Now, the second question we asked is, how plastic is the human auditory brain? We hypothesize these three things. Over time, human listeners can adapt to anatomical frequency mismatch, even when there's spectral degradation, as in the case of a cochlear implant. We also hypothesize that some listeners may not adapt completely, and that individual differences in the ability to adapt completely may have clinical implications and may be addressable by different kinds of interventions. So we're going to examine perceptual frequency mismatch as a function of location of the most apical active electrode and time after activation. Okay, I'm going to stay for a while in this slide because it shows a summary of a lot of data. Each one of these panels shows data from a group of patients, and they are sorted by depth of insertion. The upper left panel shows patients whose deepest electrode insertion is less than 350 degrees. At the other end, these are insertions greater than 600 degrees. This panel is about one turn, 350 to 400 degrees. Most cochlear patients fall here. The x-axis is a logarithmic scale showing time after activation. So the first dashed line is half a day, half a week, then 1 month, 1 year, 10 years. These lines show a local representation of the average data. And what we see, and we confirm in our statistical analysis of the data, is that there are two clear effects. Electrode insertion angle has a very clear effect. Deeper insertions result in less perceptual frequency mismatch. And there's also some change over time. But it's interesting to see how for patients with deeper insertions, on average, there isn't as much change. But to some extent, that may be because little change is needed. Their measured low frequency mismatch is limited. For patients with shallower insertions, there's some change on average, but as you can see, these are patients with 1 year or more of experience, and there's still a lot of frequency mismatch. And in these two cases, it's more intermediate. There's also a trend towards decreasing frequency mismatch, but not for everybody. And these are the three research audiologists who have had a recent major role in obtaining these complicated data. So yeah, I mentioned that deeper electrode insertions result in less perceptual frequency mismatch, and perceptual frequency mismatch tends to decrease with experience. A possible interaction effect, but that's less consistent. Now, one important practical question is, why should we as clinicians care? Is frequency mismatch associated with speech perception, or is it something that simply changes speech quality? And we find that in these patients that we tested, if we sort them in three groups, low amounts of frequency mismatch between minus 150 to 300 hertz. These are this violin plot of their AZ Bio sentence perception scores. This is for patients with high frequency mismatch, more than 1,000 hertz. And this is for patients whose perceptual low frequency mismatch is intermediate between 300 and 10,000 and 1,000 hertz. And you can see that the median is starkly different for these three. This is highly significant, 20%, 50%, 80%. So it seems that perceptual frequency mismatch is associated with speech outcomes. So in conclusion for this part, acoustic models that sound like the cochlear implant show evidence of perceptual frequency mismatch, and this is especially clear in the first few days or months after activation. The effect is more pronounced in cases of shallower electrode insertion. The good news is that the human auditory organ is plastic and it compensates for this perceptual frequency mismatch, and some patients show complete adaptation after a few months or even quicker than that. The bad news is that some listeners do not adapt completely, and this may have a negative impact on speech perception. So the question is, what can we do? So I'm going to describe three potential approaches that are experimental, not being used clinically, not extensively at this time at least. If there's a perceptual frequency mismatch, we can fix the frequency, fix the place, or hack the brain. So let me explain what we mean by that. Let's talk about fix the frequency. We have developed an iPhone app that allows patients to experience different frequency tables in an indirect way, but a good enough one. This is what it looks like on an iPhone. Let me see if I can play.
This is the screen our iPhone app uses to select frequency allocation tables, or FADs. Each square represents a different FAD.
Now you are hearing the actual acoustic output of the app. Remarkably, Cochlear Implant users can barely tell the difference between this processed signal
and the original unprocessed signal. Subjects can easily select alternative FADs in real time as they listen to running speech in any real-world environment.
So I can imagine that my good friend Bear, who's listening to this, may be wondering, Mario, you can't change the FAD using an iPhone app. And that's true. What this app does, it's a tone vocoder with fixed output tones. Each tone is centered at exactly the center frequency of each electrode in the patient's speech processor. And what we do is change the analysis filters. The patient changes them until his perception or intelligibility of speech is optimized. And these are the two terrific computer scientists who have had a major role in the development of the app. The computer scientist on the left also happens to be a trained neurologist who works in our department. So let me show you one case study where the use of the app was quite successful. This was a single-sided deaf patient who had pretty normal hearing in one ear and received an implant in the other ear. He used the standard clinical default FAD for 4 years. This frequency table starts at 188 hertz. Then he used an anatomical frequency map, quote unquote anatomical, during a 1-month experiment where the low frequency edge was changed to 688 hertz. And then he kept it for another year. He liked the quality better than the standard one. And the reason they use 688 is because this shows the electrode insertion, and this insertion angle, 414 hertz, would be closest to this frequency according to Greenwood's equation. Then a year later, he used the CA Select iOS app to select his preferred frequency map. And he's one of those patients who has a clear preference. Took a few minutes, and he picked the FAD starting at 563 hertz. This is his description of how he experienced that.
So I'm using this, and I'm, for the first time in 5 years, hearing my voice in the processor sound the same as it does in my natural ear.
Okay.
For 5 years, I haven't been able to do that.
Okay.
And this today is the very first day where I'm able to hear my voice and your voice and Nicole's voice the way they sound in my natural ear.
Yeah,
and it's kind of blowing my mind, to be honest.
That's terrific.
So
we made several acute measurements. So we measured music, sound quality, listening effort, and speech intelligibility. These are subjective ratings. And on the right, we show data from BKV sentences. And we did this with several frequency tables, starting at 188, the one he used for 4 years, and 688, the one he used for 1 year, and the one he selected that he had used for only a few minutes. And you see that the green self-selected one is as good as or better than the others in either sound quality or speech, despite the fact that he only had a few minutes of experience with it. I want to add at this point that this will not happen for every patient. Some patients try different FADs and they sound about the same and their speech perception is about the same. We don't know why that happens. I don't know whether patients could benefit from an alternative self-select FAD represent 10 or 40% of the CA population. And again, we're talking only about postlingually deafened patients. That's something that we hope to investigate. Now, we can change the frequency table, or we could change the electrode location. One way to do that is to repurpose one of the ground electrodes in the cochlear device as an additional electrode placed in the apex. And these are the principal investigators in that effort, my colleagues Tom Rowland and David Landsberger. So the basic idea is that the EC1 electrode, which is redundant because the cochlear device has another ground electrode in the case, can be repurposed for apical insertion. And our pilot studies show that this electrode results in lower frequency percepts than any of the other electrodes, and that could potentially be used to change the frequency maps. We don't know whether that enhances speech perception or not, but that's, again, something we are investigating. And last but not least, one thing we can do is to hack the brain, use neuromodulation to enhance perceptual learning and facilitate the process by which users of cochlear implants understand speech. So over the years with the Fromke lab, we have developed a rat model of cochlear implantation. This was done initially by Julia King, a terrific MD Ph.D. student. It was difficult to insert Cochlear's animal AIDS channel electrode in the rat cochlea. In fact, our colleagues at Cochlear were surprised to hear that one can do that in a rat. But Julia is an excellent surgeon, and she had terrific help from Tom Rowland, who's one of the best human cochlear implant surgeons. So what we can do with these rats are, I have learned, smart animals, much smarter than mice. We can train rats, for example, to identify one tone. What the rats do, this is before cochlear implantation. They initiate a trial by poking here, they hear a tone, and if the target's tone is presented, 4 kilohertz, then they nose poke and they receive a reward. They do not receive a reward, but a punishment for the non-target tones. The punishment is just that they have to wait longer before they can hear another tone. Animals can be trained to identify a tone and disregard the other tones. We then implant them and repeat the training with a cochlear implant, where they identify one electrode being stimulated, and they have to withhold nose poking if the non-targeted electrode is stimulated. Now, building on this model, what our student Erin Glennon did was to use neuromodulation to enhance perceptual learning of the cochlear implant task. What she did was to stimulate locus coeruleus, which is the main source of noradrenaline for the entire brain, and it's thought to modulate attention processes. So what she did was to inject transgenic rats with channel rhodopsin, so that the neurons in the locus coeruleus would be excited by presentation of light. And she also inserted fiber optic in the fourth ventricle to achieve that. We had 2 groups of rats that received paired stimulation. So the CI target tone, let's say this is electrode 4, was stimulated for 1 second together with stimulation of the locus coeruleus. And this was done for 5 to 10 minutes every day. In the sham stimulation, we used a different type of stimulation that didn't result in activation of the locus coeruleus. And what we found was interesting. So these are average results for the sham group in black and the experimental group in blue as a function of time after activation. On the y-axis, we show CI performance the parameters d prime. And conventionally, we say that d prime better than 1 shows reasonably good performance, discriminating the target electrode from the non-target electrode. So one thing we see is that performance improved more for the neuromodulated group in blue than for the others. And interestingly, when instead of looking at average, we look at the individual curves and we measure how much time it took the rats to achieve a d prime of 1, we see that the rats with neurostimulation achieved that on average in 2 days, which is remarkable. That was the case for some of the rats in the non-neuromodulated group, but many others required as long as 2 weeks to achieve the same result. This was a highly significant difference. And these are the 2 other major collaborations in the study. As I said, this was done in Rob Fromke's lab. And Ariel Haidt is the new postdoc who works in my lab as well as Rob Fromke's. And he works importantly in both animal and human experiments. Now, of course, we can't stimulate locus coeruleus in human. We cannot put a fiber optic inside the brain. We cannot, put the required virus in the locus coeruleus. But one thing we could potentially do is to use vagal nerve stimulation, which does activate locus coeruleus neurons through anatomical connections from the nucleus tractus solitarius. We are exploring in particular the feasibility of stimulating Arnold's nerve, which is a branch, the auricular branch of the vagus nerve. And again, just as in the apical electrode project, this could be done by repurposing the redundant ECE1 ground electrode from the cochlear device, except in this case, we would put it in next to Arnold's nerve instead of in the apex of the cochlea. We haven't done this in humans yet, but we're about to. We have IRB approval. This was deemed a non-significant risk study. And the study we're going to perform is simply a non-inferiority study with respect to standard clinical practice. So, in summary, this is the overall summary for the first part of the talk. Cochlear implants work so well because the human brain is adaptable even in adulthood. And again, we're talking about postlingually deaf adults for the most part. The ability to adapt has limitations that are clinically relevant, but interventions to facilitate adaptation may be possible. Okay, up to this point, this is almost the same as the lecture I presented at the American Neurotology Society back in May. But I also want to show you another data set that I think is very interesting and that seems to conflict maybe with something that I showed you before. So what is it? It's another way of measuring perceptual frequency mismatch with a more traditional method using electroacoustic pitch matching, but with a few methodological differences, hopefully improvements from what we have done in the past. Again, this was done on single-sided deaf patients, and they compared CI electrode stimulation pitch to pure tones presented to a normal ear. So we use a computer to provide stimulation to the cochlear implant site and to present an acoustic pure tone. Stimuli were loudness balanced before the experiment was done. And importantly, we ran 4 adaptive staircase tests for each procedure. 2 1 up, 1 down procedures, 1 3 down, 1 up, 1 down, 3 up. And our final result was the average of these 4 runs. Importantly, we were careful in choosing the starting frequencies. This is important because the different procedures, 1 up, 1 down, 3 up, 1 down, and so on, are expected to yield a different point in the psychometric curve. This is the putative psychometric curve for a patient. Let's say we stimulate an electrode, say electrode 22, and this is the percent of times that a given acoustic tone in the x-axis sounds lower pitched or higher pitched than the electrode. I'm not going to go into methodological details, but I want to say that if the data are well behaved, the results of the 3 down, 1 up procedure should be always a higher frequency than the 1 up, 1 down, and it should always be higher than the 1 down, 3 up. So we checked that this was indeed the case, and we're very happy to find out that 77% of the data survived this validity check. And I also want to say that the data I'm going to show you now, the results are the same, largely, whether we implement the validity check or not. So it's important to mention some methodological differences with some of the prior studies. Again, each point is the average of 4 tasks with starting points carefully chosen to reduce bias. We did a novel sanity check and analyze all data with or without the sanity check. Stimuli were loudness balanced. So first, let me say that results showed very clear tonotopicity. We always used the most apical active electrode in this experiment. And in some of the experiments, we also used other electrodes, the most basal and 2 intermediate ones. These data for the most, these data for all electrodes. The angle of insertion is in the x-axis, the pitch match frequency is in the y-axis. So you can see the data are noisy, but there's a clear trend towards tonotopicity. The deeper the insertion, the lower the pitch match frequency. Here in red, we show the Greenwood function, and in blue, we show the NYU function that, as I said, is 1/3, 1/2 an octave different from Green. Almost all the data points were below both the Greenwood function and the NYU function, with the exception of these. These are worthy of a separate conversation. I think that has to do with the fact that they are fine structure channels in MED-EL patients. But let's disregard that for a moment. So these are basically the raw data, pitch matching as a function of time since initial stimulation for 4 different electrodes for a variety of different patients. The most apical electrode, which is lower frequency, the most basal electrode, which is higher, and 2 intermediate ones. So you can see very clearly that there's an influence of electrode location. These points are lower, these are a bit higher, and the highest. However, what we don't see very much of, and there was not a statistically significant result, is change over time. This was a surprise to us because we did see that change in the speech-based estimate of perceptual frequency mismatch. And some studies, but not all studies, had found changes as in pitch matching, electroacoustic pitch matching as a function of time. One of the interesting exceptions, of course, is Vermeer and Landsberger, who tested MED-EL patients. This is another graph. Instead of showing the raw pitch match frequency, I'm showing the frequency mismatch for each electrode. And again, there's no significant change in frequency mismatch over time. There's a few subjects who do show a change initially, but the big picture is lack of change. I'm going to show you the same data separately for each electrode. In this graph, blue is the most apical electrode, purple is the most basal. Let me show the 4 separately. And yeah, so one thing that we found that's completely consistent with a speech-based estimate of frequency mismatch is that for the apical electrodes, the pitch matching frequencies were consistently higher than the clinically assigned center frequencies. That's why the frequency mismatch is positive, 1 or even 2 octaves. And that doesn't seem to get a lot better over time. So these electroacoustic pitch matching data confirm several patterns congruent with prior literature. There's a very clear, strong tonotopic relationship between electrolocation and the corresponding pitch matching scores. It seems that the perceived pitch appears to be overestimated by frequency police functions in the literature. And the results were consistent regardless of whether we used the whole data set or only the post-sanity check data, which underscores the robustness of the findings. Now, the thing is that, as I said, electroacoustic pitch matching data showed marked stability over time following initial stimulation. And this is in contrast to prior studies showing changes in electroacoustic pitch matching over time. Patient reports, you talk to a patient, they say, yeah, talking to my wife, she sounded very funny initially like Minnie Mouse, but now it's a lot better and I can understand her better. So that speaks to changes in perceived pitch over time, at least in response to speech. And in fact, we measured that quantitatively in the first data set that I showed you with the selection of the acoustic models. So this makes me wonder, is it possible that the pitch of perceived pitch changes with experience for speech changes? I need to change this sentence. Well, the pitch in response to single electrode stimulation stays largely unchanged. In other words, the patient might say, yeah, my wife doesn't sound like Minnie Mouse anymore. She sounds like a normal woman. But yeah, electrode 10 or whatever still sounds very high pitched. So, that's a question that I haven't answered. And some of our next steps will be a more fine-grained analysis of possible changes in the days after initial stimulation, try to obtain more temporally detailed longitudinal data, and explore the possible distinction in adaptation to perceptual frequency mismatch when using speech stimuli versus pure tones. And of course, the good news is that we have many principled potential interventions to address frequency mismatch in clinical CI populations. These interventions have not been translated yet to the level where they can be used routinely. The one I would say is the closest would be the use of the CI Select app, which is available for free in the App Store. If any of you are interested in using it, please let me know and I'll send you a copy of the manual that we put together. So now that's about it.